Hello, Eugene Aiello here for our next video in my series. And if you followed me along this far in the series, I'm happy you did. Reminder, Conscious Canary. This book is what this video series is discussing. I'm briefly going over, and I want to say that again briefly. I'm trying to make it simple in little bits and pieces for people to follow, or people who do not have a lot of background, so maybe they can get through this book a little easier. A lot of my patients were very interested in this. So this video, probably my most exciting video, has very little to do with what I do in practice, but when I was trying to discover what was occurring with those cold lasers in neurology, I started developing this biophotonic model. And it began early on with embryology, trying to figure out what that connection was. As we discussed the glial cells, it was a big breakthrough to me when I found out that the melanocytes and the Langerhans cells were all embryologic derivatives of the glial cells. As I started working through this network, that I'm getting to the point where there has to be a cellular connection. Each individual cell in itself is its own biophotonic system. So a single-celled organism is going to be accepting all the biophotonic frequencies directly through its cell into its microtubule system. When we get into multicellular organisms, every different cell is going to have a very specific function within that organism. The glial system is going to be controlling the entire organism, but each individual cell is going to have some ability to receive a message, receive a certain combination of frequencies, which dictates how that microtubule system within that cell is going to behave, that relates to the nature of that cell, the genetic expression, how that cell is functioning. Early on in my research, I think I mentioned earlier with, maybe I did in another video, but anyway, I was doing a lot of research on how frequencies are accepted. So looking at just basic telecommunications, how do our cell phones work? How does AM/FM radio work? What is the mechanics of electromagnetic energy used for in telecommunication industry, and how do these different things function? I had to spend a lot of time learning about antennas, and I briefly spoke about the concept of a dipole antenna. A dipole antenna is going to have two parts to it. It's going to have an active receiver part of the antenna and then a ground. So this active receiver part of the antenna has to have a quarter of the wavelength of which it's accepting. So when I was looking at the cell structure, I was pretty blown away when I saw that centriole at the center of that microtubule system. So in quantum biology, most of the people in this field are already talking about the microtubule system emitting light, and frequencies of data somehow are interpreted. The quantum nature of the cell is within those microtubule systems. When I was looking at the microtubule system, I couldn't believe when I saw this. I talked about it earlier. The centrosome, which is composed of those two centrioles, and then there's all these interconnecting fibers in between. And there's even microtubules that will emit from this area. Microtubules come off of these two appendages in the mother centriole. So as we discussed earlier, the mother and the daughter centriole make up this unit. The mother is extremely complex. The daughter is very basic. The daughter is not going to get complexity until mitosis, when these guys split, and this will become the new mother for the new cell that's being produced. So when I saw this, I couldn't help but to think, man, that sure does look like it could be a dipole antenna. So just the basics of it, I thought, well, what's the length of these? How many nanometers are these? Well, it just so happens that these things are about 500 nanometers long. 500 nanometers times 4 would put us up at 2,000 nanometers. Well, that's right at that near infrared spectrum. So this could act as an antenna to receive frequencies from the far end of near infrared all the way down to all the visible light colors. So quite amazed. Even I'm not going to discuss it in any of this series, but there's also a possibility that we even get through UV. UV light could be very important in this model. The only reason I still keep that in my mind, but in my book and in my theory at this point, I don't mention UV light, but when I was talking about the onion guy, Dr. Dershowitz, his experiments with mitosis, that UV light seemed to be what was inducing mitogenic activity in those cells of the onion roots. So just a side note, we're going to throw that out there. So when I started looking closer, I found that this mother centriole has a distal and a subdistal appendage, almost as though you can see that the location of this would change where in the near-infrared spectrum this different microtubule antenna would work. The subdistal appendage, same thing, it's down lower. That would probably orient to the visible light spectrum. As I go a little further in my research, it was like everything was falling into place. When you look at a centriole, the cross-section of a centriole looks like this. So the centriole is composed of 9 units of 3 microtubules. So there's 3 microtubules making a bundle. I refer to these as rods in my book. So we got 9 rods composed of triplet microtubules. Well, already at that point in my research, I was already considering red, green, and blue were very important in the biofrequency system. So I looked at that and thought, oh my goodness, that kind of fits. Going back, every one of these centrioles, these triplets, all of these triplets have a subdistal appendage to them. One of these triplets is going to have a distal. So you got all 3 of them could be composing those red, green, and blue, and one of these 3 is going to be accepting near-infrared frequencies. So what I started thinking is, this is going to be a long leap from there, but embryology, as I said, very important in my model. I was basing everything off of embryologic connection between the systems. I did a little bit of looking into stem cells, and I was reading how stem cells, we start with the one fertilized egg. That fertilized egg goes into 3 stem cell lines: ectoderm, mesoderm, endoderm. Very big when I was first researching my model, because the ectoderm creates the skin and nervous system. So right away, that connection of the pigment system to the nervous system was just a small connection, but it was enough I started following it. The other one becomes the mesoderm. The mesoderm is your musculoskeletal system, your cardiovascular system, your renal system, all your blood cells, your lymphatics too. Your endoderm is going to become your gut, basically anything that's related to digestive. So looking at this concept, I started thinking, well, basically the whole process of differentiation, stem cell differentiation, probably has to do with how these different rods are linked up. We have 9 rods. As we go further in stem cells, these 3 stem cells will break into 3 new lines themselves. That's making up 9 final stem cells at the end. After those 9 stem cells, all differentiated adult cells come from that 9 stem cell line. Well, I'm looking at the 9. The 9 makes sense to me. So I started theorizing that these interconnecting fibers are connecting the mother to the daughter. So every process of differentiation, the first beginning process of stem cell differentiation has to do with what's linked and what's not. So my theory, and it's still my theory and it's pretty solid, that I think that the first process, when we go from that fertilized egg, we're talking that guy that guy has 9 of the rods connected. Meaning this cell can become any cell, any adult cell. It has the possibility of receiving any one of those combinations of frequencies. When we get down to this, now we're going to say, okay, we lost 6 connections. So when you're talking about that 3 germ layers, those 3 stem cell lines, each one of those is going to keep 3 of these unit rods still linked up. As we go down to the final, that's going to be 1 of these. So the process of stem cell maturation is basically just a disconnecting of those different connections between the rods. Now, why is this important? I believe that each one of these guys has a different preset location. So if we're going to talk about the red rod, if we have a subdistal appendage, it could be connected between 187.5 and 155, and that would all be within red frequency absorption based off the quarter of the wavelength. So if you multiply those out, you're going to be within the red spectrum of light. That's 32 nanometers distance between the two. So my belief is that we have a preset where, say on this first junction, maybe that one has a high preset on it, okay, versus medium or low. So there's 3 different presets that occur within this system. Then we get to the green. The green can be at 142.5 nanometers to 122.75. That's another 20 nanometer distance. So this again will have 3 preset options. So high, medium, and low is what I describe it as. Same with the red, high, medium, low. And then when we get into this blue, we could be at 122.75, 112.75 nanometers. That would include all the blue light spectrum, which is only 10 different nanometer wavelength. So I came up with it took me forever. I came up with different combinations of how we would get the 9 different groups of preset arrangements. So I came up with, okay, when we're talking about those first 3 stem cell lines, you're going to have a combination where you got a preset of red high, green high, just blue there. Red high, green medium, blue there. So hopefully you're following this. I went through and figured out all the different combinations of how those subdistal appendages could be linked to this system. That makes up 9 different presets. So that makes up our 9 different stem cell lines. Hopefully you followed that. I'll give it a second. So preset rods, basically, with 9 different combinations. So this guy here has all 9 of those combinations of preset rods set up. When we start dividing it out, all we're doing is disconnecting the mother and daughter centriole dipole connection. So these guys have all the possibilities. It's a matter of if these dipoles are linked. When we get to this last final stage, only one of those is linked. So we've got 9 different dipoles with 9 different preset possibilities. So how do we go from stem cell, 9 stem cells, down to every different adult cell? Oh, if you start reading about embryology and how differentiation occurs, currently conventional science has not figured it out, at least to my liking. They do know that the notochord, so when the embryo is developing, there's a process of invagination of that ectoderm, that neurologic part of the ectoderm, into the mesoderm. And that mesoderm, in the mesoderm, there's a structure called a notochord, which is on the ventral aspect, the front aspect of that primitive brain and spinal cord that just invaginated into the mesoderm. Well, that notochord, they know that that is inducing differentiation. That notochord is guiding the complete development of that embryo into a fetus. So the way they know that is if they destroy part of a notochord, parts won't develop. So they know that's the master control center. I believe that when we have this stem cell differentiation going on and these stem cells are dividing, this original master cell, which I mentioned earlier, master cell up in those astrocytes, I think that this keeps dividing and these are producing that notochord stem cell line. This cell is able to receive and send all the signals to the rest of the cell. Eventually, that notochord is going to start working through the glial system as the embryo develops in fetal stages. And I believe at the end, that central glial system is still retaining some of these master cells. That's the quantum computer up in the brain. That's receiving and sending that coherent signal I keep talking about. Another thing I want to note, when we're going through these different stem cell differentiations, none of these guys have active mitochondria. It's not until they become adult differentiated cells that the mitochondria turn on. So basically, once this dipole antenna system is completely set up and modified, that mitochondria turns on because now that cell is part of that organism. That cell is receiving the message. So it has a function at that point. Prior to that, no function. So going back, when I brought up the notochord, conventional science has only come up with a protein called a hedgehog protein. Excuse me. That hedgehog protein is responsible for differentiating all the cells according to conventional science. So that notochord is producing hedgehog proteins, and that protein is telling each one of these stem cell lines what to become. They are not off because they know that if they disrupt hedgehog proteins, that's another way to delay development of that embryo. I believe that that hedgehog protein is working not by is not working by lock and key mechanisms or some type of chemical cascade. I believe that that hedgehog protein is interacting with transmembrane bound receptors that are linked to that microtubule system that's coming in and attaching at these distal and subdistal appendages. I believe that that hedgehog protein is being emitted with a very specific frequency. And when it when it sends its biophotonic frequency when it's nearby, because it's very important, location matters. Those hedgehog proteins have to be very close in proximity to those cells to do that final stage of differentiation. It's going to send that biophotonic signal in there, and I call it the fine tuning. So you got those 9 presets that I discussed here that are all located on each individual one of these rods. That hedgehog protein is fine tuning that to tell what that cell is going to become in the end. That final end, I'm sorry, I should start on that final end product adult cell is basically absorbing that coherent signal in a certain combination of those 3 frequencies, including near infrared, and that is what's going into that quantum system within that microtubule system. That's what's telling the cell what it's going to be and how it's going to function. It's forever going to absorb that specific combination of frequencies. So, quite a mouthful there. I think I covered all I wanted to in here. We are going to discuss at some point what cancer is, because cancer is going to be a very interesting part of this biophotonic system. Cancer cells are adult cells that lost that message. So they revert back to being a stem cell, kind of, pretty close. A cancer cell no longer has a mitochondria turned on. Cancer cells are only using glycolysis for energy consumption. So it's very obvious to me that cancer is a process of this signal not being received, and these adult cells are going backwards. The other conventional theory of cancer lies within the populations of these stem cells that are within the organ system. So every organ still is retaining some of those 9 stem cell lines, obviously for repair. Another theory in cancer is that those stem cell lines are the beginning of the cancer cell. Hard to say. I'm not an oncologist. It's not my field, but it's very interesting though that cancer seems to be some disruption within this ability of the cell to receive its message. When it doesn't have the message, it's no longer part of the organism. So a topic on that's kind of cool that I found is cancer cells, and I think it's the hema, whatever, I can't remember the line that they study. I believe it's a uterine cancer or cervical cancer cell. That cervical cancer cell is the same cell line that they've been using for years. Meaning when you take a cancer cell or a stem cell out of the body, you can keep it alive. You can keep it alive indefinitely. If you take out another cell and you take an adult differentiated cell out of the body, it's not going to live very long. It's not getting that message. Its mitochondria aren't functioning anymore. So pretty interesting when you start connecting the dots here. And I was pretty blown away when I started piecing everything together. And granted, this is my theory. You know, would they ever be able to see that when they start studying different cell types, adult cell types, that they're seeing that these subdistal appendages have very different locations, and that location seems to be consistent with same cells have same locations, different cells. A kidney cell is going to have a whole different setup than that liver cell. So pretty interesting. I think that the receipts of my theory could be discovered by studying those centrioles and where those subdistal appendages attach. So I think that's all I had for this. I threw a lot out pretty quick. I don't think I skipped anything because in reality, it's really not that complicated. Once I started looking at it, it just completely made sense. I guess I could add this. The variability is allowed within the system. What I mean by that is when we go through that fine-tuning system, when you're moving that up and down, so many possibilities of different wavelengths. Especially because I believe from the quantum input, so those inputs coming from the pigment system I discussed, that variability isn't going to be as simple as one of them 635 nanometers. I think we're going down multiple decimal points of specificity. I can't remember the number that I came up with, but the number I came up with was just possible. There's so many other factors that can fit in, but you're talking that much specificity within that final tuning process. That's why there's such a large variability in how many adult cells we can have. It also allows for the large variability of when I said that different electromagnetic pollution is affecting that quantum input, it's shifting that slightly off tune. Well, the amount that it's shifting off tune, we're getting into these back end of those decimal points. So these cells are still going to be able to function. You're not going to have a collapse decoherence of the system at the quantum state. You're just going to have slight inefficiency. Those cells are just not going to be pushing out those frequencies within the microtubules as they should. Most of it should function, but obviously disease processes occur, and I believe the electromagnetic system is the root of it. So if there's more to add, I will add in the next videos. Thank you for watching, and again, I hope you're enjoying my series.